is an outline of the content. We'll start by defining what cytogenetics is. We'll look into also the different types of chromosomal aberrations linked to reproductive issues. We'll also look into techniques, applications, and also examples. All right. So what is cytogenetics? So cytogenetics is, by definition, is just the study of chromosomes. It could be the structure, number, so the numerical or the structural aspect of chromosomes. This is the branch of study that deals with the chromosome structure and number at the cellular level. So if in a broad sense, if you talk about veterinary cytogenetics, then it refers to the cytogenetics that's applicable to veterinary medicine, which is related to the genetic issues with animals. So generally, we need to have a very complete and balanced set of chromosomes required for a normal development of an individual before born or also after birth, meaning prenatal as well as postnatal. But if there is something wrong with this complete and balanced set of chromosomes, then it leads to chromosomal errors, leading to problems like consequences, which can be related to the reproductive performance or fertility due to failure of production of viable gametes or due to early embryonic loss. So sometimes when you have a chromosomal aberration, so the gametes produced may not be able to successfully fertilize to each other. And sometimes even if they are able to fertilize, after the zygote is developed and the embryo development starts, then the embryo will not be able to continue further during the pregnancy period, and it will be lost. So I think a lot of embryonic loss occurs during the very early stage of the pregnancy period, which is undetected and is believed to be one of the major problems, especially like in dairy cattle as well. So this fertility problems, if it keeps on going, it does have a very big impact on the economy of the farm or the production of the farm animals, especially in animals, in livestock animals. So the other way is if the embryo succeeds to survive, even though it's resulted from the chromosomal abnormality, and continue to finish up with the gestation period, the pregnancy period, then there will be still a problem after delivery or after birth. So a postnatal developmental defect will occur, or in other words, maybe if there is no any obvious developmental defect, the animal might continue as a carrier of that that chromosomal defect and will transfer that defect into subsequent generations. So these are the consequences whenever you have chromosomal abnormalities. So we mentioned about cytogenetics, studying about the structure and number. So when you talk about chromosomal abnormalities, we are talking about abnormalities either related to number, which is a numerical error, or it could be related to structural rearrangement or structural abnormality. So you may classify into two. We'll see the details later. And when we talk about the complete set of chromosomes, like a diploid number of chromosomes in cattle, for example, when we talk about 60, it consists of the autosomal chromosomes and also the sex chromosome. So sex chromosome is always a pair, that is 2. The remaining ones are autosomal chromosomes. So we have like 58 autosomal and 2 sex chromosomes. So that's the normal set of chromosomes in cattle, for example. And human, you have go 46, 23 pairs, so 1 pair is for sex chromosome. The remaining are considered as autosomal chromosomes. This chromosomal abnormality or the imbalance or the aberration, it can happen either during the gamete formation or during the cell cycle that involves gametogenesis, or it can happen even after the gamete is formed during fertilization. So these are the points of reproductive cycle where you can have those aberrations or chromosomal abnormalities. So this diagram shows you a normal gametogenesis for both sex cells, for spermatozoa as well as for oogonia, for the ovum. So you can see the meiotic cell cycle, the meiotic division. Initially mitosis, you have a full set chromosome, then there will be multiplication by mitosis, then they will undergo division, and at this stage there will be segregation of the chromosomes, as you can see here. So they will go to separate cells. So sometimes it occurs like here. Instead of segregating equally, a bigger portion will go here, maybe this one becomes 0. So this one is known as like non-disjunction problem, or maybe it occurs at this stage. Instead of separating into two, reduced with 2 chromosome numbers here, maybe this one becomes 0 and the whole goes to one side only. Again, here also, if it does happen, then that will result in imbalance, and it will result in numerical error. That's a numerical chromosomal aberration. But normally it has to go down like this, and end result will be haploid chromosome number compared to the parent cell, right? Which is N. If you have 2 N here, lastly you have N. Same for oocyte, for the oogonium or genesis. Initially you'll have a diploid number here, become haploid. So all gametes are haploid, as you know. This is an example for you to understand what I mean by this non-disjunction of the chromosomes during the cell cycle and how it leads to problems. So you have a parent cell here having a diploid number, all right? You have 4 here, 4 here. So during division, in a normal cell division process, half will go one side, half will be another side, right? So this is one of, for example, the ova or the sperm with a balanced chromosome number. But on the other side here, as you can see, the parent having 4, at this stage during division, supposed to have one from here going this direction, another pair to go this direction. But what happened here is these two go together with one of them here in one side to another cell. And only one has shifted or separated to go to the other cell. So at the end of the day, you have already a numerical error here, an unbalanced chromosome number of these gametes. So when these two gametes, let's say this is a sperm or ova, when it unites or during fertilization with another gamete having a balanced chromosome number like this, it will end result with this kind of zygotes having a chromosomal constituent of numerical error. So here, if you combine these two, what will happen is you will have 3 chromosomes here, right? Which is 1 chromosome less the expected normal chromosome constituent. So in this case, it's called like monosomy because of the reduction in 1 chromosome number. So this is a human chromosome example. Instead of 46, you will have 45 in this case. And if these are the gametes involved during fertilization, these two, here you have a normal chromosome constituent, but here there is extra chromosome number, right? So when these two join, they will end up resulting with a chromosome number which is surplus, excess, all right? So these smaller chromosomes become triple instead of supposed to be 2, which is the one missing here, right? Actually is here. So giving extra chromosome number to this group, and this is leading to trisomy of this particular chromosome. So probably you know that this trisomy of 21, chromosome number 21 in human, that leads to a Down syndrome is one of the common congenital disease problem. It happens when this kind of chromosomal rearrangement occurs. So just to give you a little bit more on the basic aspect of the chromosomes, how morphologically the chromosomes are classified. We actually examine the chromosomes because I say cytogenetics is a study of chromosomes at cellular level. So we have to obtain the chromosomes from the cells during the cell cycle. So the best time for you to observe the chromosomes clearly is at the metaphase stage of the cell cycle. Metaphase stage of the cell cycle. So you need to remember the basic cycle involving the different stages of the division process. So at metaphase chromosome, this is how you will expect to see it. So this is the appearance, all right? So this is a photograph. So you have got this chromosome, sister chromatids, all right, one, another one. They join together with this centromere. This is a structure that joins the 2 sister chromatids together. And those arms above the centromere, they are referred to be the short arms, and below the centromere, they are long arms, okay? And based on the location of the centromere, these chromosomes are also classified into different types of chromosomes. When you have at the middle, the centromere is located in the middle, so you have a metacentric type of chromosome. When it is a little bit away from the middle towards the end, we call it a submetacentric. And when the centromere is located towards the end or at tip, we call it this acrocentric and telocentric type of chromosomes. This morphological classification is important in differentiating chromosomes of different species of animals. You will see that later on. So now let's talk a bit about the techniques. So what is this cytogenetic and how it is conducted? So generally, we have got these two general classifications of cytogenetic technique. One is conventional, another one is molecular cytogenetics. And this conventional technique is the most commonly applied technique in veterinary cytogenetics. All right. So here in conventional technique, you will be utilizing gene sustaining of those metaphase spread chromosomes from the cell. All right. And apart from that, when you want to see more detailed aspects of the chromosomes, you will be doing these banding techniques. We have what we call this banding techniques. There's a G banding, C banding, R banding, and so on. There are many different banding techniques that can be applied on those metaphase spread chromosomes to differentiate the numbers and also the structures, and if there are problems. And the molecular aspect, this actually involves a use of chromosome-specific DNA probes, all right, specific to each of the chromosomes. So these days, commercially, there are different types of chromosomal DNA probes available, so you have to utilize that. And one of the common technique under this molecular cytogenetics is the fluorescent in situ hybridization technique, which is abbreviated as FISH. So we have monocolor, multicolor, or spectral karyotype type of the FISH technique. So you can see the chromosomes differentiated by different fluorescent producing colors based on the probes that you use during the procedure. All right, so let's talk a bit more on this conventional technique. So as I said, this is a most commonly applied technique in animals. All right, and when you look at the steps and the procedures involved, it all begins with collection of the sample, and the best sample usually is the blood sample. So you have to collect this sample aseptically to avoid any chance of contamination. And then after you harvest the blood sample, you need to go for cell culture, all right? You need to go for the cell culture and chromosome harvesting. And after you harvest the chromosomes, then you can go for staining, banding, and microscopic examination. So very important, especially for those students attending this presentation. During your sampling, you need to make sure you do it aseptically. You clean very well the area of your sampling at the skin. If you are collecting from the jugular vein, you have to make sure very well disinfected the area, all right? And then you have to collect the semen aseptically. So you try to get multiple samples, like 2-3 vacuum tubes, collection tubes, so that you can increase the chance of getting a pure culture at the end of the day. So once you get the sample, the blood sample, you have to centrifuge to separate the buffy coat layer. This is the layer which is important for you to culture, because we need the monocytes and those lymphocytes for the culture. They are the ones to be induced to multiply during the cell cycle, all right? So you can either remove the plasma, or you can just directly go into the sample and aspirate the buffy coat layer here with the lymphocytes. So that is the sample to be used for the culture. And here is the whole protocol in detail given, all right? So the cell culture will be established using the sample that you have collected, the buffy coat layer. Then after that, you have to add the colsemi. There is a mitogen to be included to initiate the multiplication of cells. And then this is usually a culture that will be lasting for about 3 days, 72 hours. So a few hours before the end, you have to add this colsemid, which is important to stop the mitotic division process. So that's mitotic arrest, okay? Then after that, you have to add some chemicals to, you need to centrifuge, separate it, and then add some hypotonic solutions usually KCl, potassium chloride. And then this hypotonic solution will help to swell the cells, all right? And it will help to disperse, or the cell to burst and release the genetic component, that is the chromosomes, from the cell. So you keep centrifuging and doing it multiple times here, and you add a fixative into the solution to fixes the chromosomes. And after that, you prepare your slide, all right? And then you drop the sample. So you have to do it in a higher, a higher level to disperse the chromosomes as they drop on the slide. And after that, you do your staining, like the GIMSA staining I mentioned to you, and go directly to examine under microscope, or you do the banding techniques, all right? And then you go for microscopic examination. So looking at the application using this conventional GIMSA stain, this is just an example from my previous work, and you can see well-spreaded chromosomes, metaphase spread chromosomes from a Frisian Sahiwal cattle. So you can see the sex chromosomes are well differentiated. Remember the morphological classification of chromosome. So this is a submetacentric type of chromosome, which is a sex chromosome. So you can easily differentiate. This is a female cattle with XX chromosomes. The rest are the autosomal chromosomes. And this is after karyotyping. Karyotyping is arranging the chromosomes in order according to there is a system developed by the International Society for Chromosomal Nomenclature. So you have to arrange the chromosomes like that, and the sex chromosome will be at the end. The autosomes will be arranged in a decreasing order in the size. This is the banding technique, one of the banding method, which is the G-banding. So you can see there is a pattern like a barcode kind of appearance, right? So this will enable you to pair the sister chromatids together, the pair of chromosomes with similar pattern. So the banding technique is very important when it comes to karyotyping, and also when it comes to detecting rearrangements and errors or chromosomal abnormalities. This is another type of banding technique, which is known as the C-banding. It's targeting mainly the centromere. The centromere, this is highly stained part of the chromosome. So remember, in cattle, we have got all these autosomal chromosomes having the centromere located at the end. So it is acrocentric type of chromosome that we have. Only the sex chromosomes are different, having the submetacentric and acrocentric location of the centromere. So that is about conventional. For molecular cytogenetics, I have mentioned the technique, which is FISH, the fluorescent in situ hybridization, one of the technique. And this technique is actually developed to overcome the limitation that comes together with the banding techniques, especially when you want to detect deletions, those very minute kind of chromosomal aberrations. Then you will have difficulties to get them detected using the banding techniques, the conventional method. So you will need to get these molecular techniques to detect those kind of abnormalities. So here in molecular techniques, we have got FISH, the fluorescent in situ hybridization. And as I mentioned, you have to utilize chromosome-specific DNA probes for the chromosome number that you are interested with, so that you can be able to easily fluoresce, and you can examine by fluorescent microscope to detect whether that the problem is there or not. So these are the classification of the FISH probes commercially available. You can get the centromere specific probe, targeting the centromere part. You can also get whole chromosome painting probes, the entire chromosome length. And there are single copy, that's locus specific, spectral karyotype, and also translocation fusion probes. And this is how the FISH works, the procedure, all right? So you have got different steps in the process. So you have first the metaphase chromosome spread, just similar to what I mentioned before, how you prepare from the blood sample. After you have that metaphase chromosome spread, then the next step is you will be doing labeling. So the labeling will be like those chromosome-specific probes will be labeled together, and then they will be mixed with those chromosome metaphase spreads. And at this stage, you will be doing some denaturing and hybridization technique. So you denature the chromosomes and then you hybridize it with the labeled probes that you have got commercially. And then you do some washing and removal of excess probes during the process. At the end of the day, you will have that target probe being part of that chromosome and then be fluorescing under the microscope during the examination if it is positive for that particular probe. Okay, so there are some additional advanced techniques within the molecular cytogenetic category. Spectral karyotyping, you can use multiple color, meaning multiple probes for different chromosomes at a time, can be utilized, all right? And then we have this comparative genomic hybridization, micronucleus assay, and so forth. And if you would like to see details, there's a review paper from our unit done by Yahya in 2019. Maybe you can refer to that. Quickly moving on to the application or indications. When do we need the cytogenetic work? I think this is very important as veterinarians and also as clinicians, right? So the first indication is when you want to differentiate between species, all right? Between breeds of animals also. For example, you have got a river buffalo and you have a swamp buffalo having different chromosome composition. So you have to use cytogenetics to differentiate. And the species, obviously, between different species, we have a very huge difference. So you can use cytogenetics to detect this. And the second one is you want to determine the sex of a given individual or animal. So whether it's a female or a male or a mixed or mosaic or chimera, you know, the what you call the free Martin, having both male and female sex chromosomes. You can use cytogenetics to diagnose and confirm those kind of sex differences. And then we have the prenatal genetic diagnosis, PGD, and this is usually conducted in human cytogenetic labs. And we have also diagnosis of conditions, especially like reproductive problems associated with the chromosomal abnormalities. In this case, also cytogenetic plays a lot of importance. And that's why sometimes I refer to it as reproductive cytogenetics. And that this kind of chromosomal abnormalities that you could have includes, you know, numerical errors such as euploidy. This is alteration in number. Also aneuploidy, all right, having less or extra chromosome number for a given pair. And then you have also structural abnormalities, including translocation, inversion, deletion, and also chimerism, which is an example is free Martins. And also mosaicism is another indication you can use cytogenetics to diagnose or to investigate. This is just a schematic diagram that shows the various types of chromosomal abnormalities. Here from the left side, you have got the numerical abnormalities, euploidy and aneuploidy, all right? Euploidy usually occurs during IVF, you know, having polyploidy and things like that. Aneuploidy, it can happen using autosomal chromosomes like trisomy, monosomy. Structural, it could be balanced or unbalanced. When it is balanced, usually you can have still new offspring, but as I mentioned before, subsequent production may be difficult. The unbalanced ones are always problematic. You may not succeed with breeding, all right? And this is a baseline data for everyone to remember. I think everyone knows about it. So this is the normal chromosomal nomenclature for the various species of animals that we have. For example, in cattle, Bos taurus, indicus, we have got 60 diploid number chromosome. Some metacentric, metas we don't have. We have only acrocentric autosomal chromosomes, but sex chromosomes, some metacentric for both in Taurus. And you differentiate the sex chromosome from indicus by looking at the sex chromosome of Y, which is acrocentric in morphology. Same thing like sheep, you have got goats, buffalo, horse, pig, dogs, and cats, having different morphological characteristics. All right. So this is the example that I showed you before for differentiating the sex, one of the indication. So it's very easy to identify the sex chromosomes having a different morphological appearance, which is submetacentric, compared to the rest of the autosomes, which are acrocentric. All right. And here again, a different, this is indicus. The previous one was Taurus, female cow. This is IKK Kultam bull. And this is a both Taurus. You can see the sex chromosome for Taurus, the Y1, is not acrocentric, unlike Indicus. This is like a metacentric or some metacentric chromosome. And this is for a human chromosome, XX and XY. You can see it's totally different from cattle, what we have seen. A lot of them having a metacentric autosomal chromosomes. Some of them even looking metacentric, with centromere located in the middle. And here is another indication for cytogenetics, which is a prenatal genetic diagnosis. And as I mentioned, this is commonly applied in human cytogenetic labs for detection of abnormalities in pre-implantation embryos. Those people work in IVF labs, they do this test from the embryonic tissue samples or maybe from the growing fetus as well. And this is an example of banding from conventional cytogenic technique, detecting a trisomy of chromosome number 18 in human. So this is trisomy, chromosome number 18. And this is a fish test showing the trisomy chromosome number 21, the common congenital abnormality in human, which is a Down syndrome. So you can use a chromosome-specific probes for chromosome number 21. And then you can use this fish technique to detect from the amniotic fluid samples whether this growing baby is having this problem or not, the trisomy. And this is another indication, particularly in animals, all right, to diagnose reproductive problems associated with the chromosome abnormalities. And this is one example, which is known as the translocation. So translocation, meaning that when two chromosomes fuse together. So we have one acrocentric chromosome, the first pair here. Another pair is here 29. This is for cattle chromosome. When these two join together, they created this kind of chromosome. So this is called a centric fusion translocation, because they fuse at their using their centromere. So and result is not actually normal in cattle chromosome karyotype. So this translocation was first detected by the person named Robertson. And because of that, the name of this translocation also is known as Robertsonian translocation 129, chromosome number 1 in 29. And this has resulted in reduction in fertility in cattle. And there are several reports after the first report from Sweden, from different cattle breeds across the globe. And you can see here, this is a kind of summarized kind of reports from the different parts of the world. So highest rate of prevalence of this translocation was reported from Italy, even though the number of animals investigated was little. So from this particular breed of cattle, 32% of this translocation. Also from France, they have reported from these breeds of animals, including Limousin. I think Limousin is famous around this area. They also have detected this translocation from this animal. From Sweden, as I mentioned, I think that's where the first time reported. They have also a big percentage of incidence of this abnormality from their cattle breeds. All right. And fortunately, the very famous dairy cattle breed, Holstein Friesian, has been reported to be free during that time. From Australia, Britain, they also reported 0. Also from Germany, no, from US, also reported 0% incidence of this translocation. In other species of animals, like in horses, this is an example of a report from horses, different types of translocation, not necessarily between 1 and 29. So these are the different types of translocation. Translocation even can happen between autosomal chromosome and a sex chromosome. So, and here, as I mentioned, those chromosomal abnormalities can have a very big impact on the fertility. And these are the reproductive problems that were faced with these individual animals detected with this translocation. So REAL stands for recurrent embryonic, early embryonic lethality, or early embryonic loss. So you can see many of those female animals with these problems have been detected to have this real problem. Subfertility is another problem. Congenital abnormalities after birth, also azoospermia and having small testes, is also another problem linked together with this chromosomal abnormality translocation in the horses. And this has been reported based on these conventional banding techniques of all these different types. And here is again how the sex chromosome aneuploidy can be detected. This is just pertaining the sex chromosomes, abnormality related to sex chromosome. So you can see here, there is a monosomy here, lack of one sex chromosome, only single chromosome is there. And we have a mosaic of a monosomy and another complete set of X chromosome. And you can see a multiple of different combination of mosaic of chromosomes, sex chromosomes, in different species of animals, all right? You can see the list, and resulting in a very important reproductive problems. As you can see here, intersexuality, ovarian hypoplasia. I try to summarize it here. So what you see in those females is intersexuality, ovarian hypoplasia, also infertility. In the case of males, also intersexuality, testicular hypoplasia, and cryptorchidism, also reported associated with this sex chromosome aneuploidy. All right, this is again a sex chromosome aneuploidy with more detailed clinical manifestations reported. So like, for example, with X monosomy, usually the external genitalia will look like normal, but you will have abnormal estrus with no clinical signs of estrus, or small or hypoplastic ovaries, meaning that animal is sterile, cannot be able to breed. So the highest incidence is in horses, followed by cat and dogs, cattle and pigs. So likewise, you have this trisomy also, sex chromosome trisomy, not the Down syndrome. This is for the sex chromosome. So again, externally, genitalia may look like normal, but again, these animals are infertile. And that's already reported in dogs and horses. XXY trisomy, this is what we call the Klinefelter syndrome, all right? And in this kind of animals, you will see small hypoplastic testes, abnormal spermatogenesis, azoospermia, oligospermia, and definitely these animals are sterile. And it has been reported in cat and cattle more than dogs and horses and pigs. Okay, this is specifically in bovids, all right? This is a report in bovids specifically with XXY syndrome, which is a Klinefelter syndrome. And it can happen mosaic with other chromosomal aberrations. And this is the phenotype, what you see in cattle having this kind of abnormalities. So largely you have got testicular hypoplasia. Most common for most of the animals is testicular hypoplasia. In some species, you might see some additional problems like azoospermia, all right? Azoospermia in bulls. Also in buffalo, testicular hypoplasia, sheep and goats as well reported. So in conclusion, just for a take-home kind of message, from all this presentation, you might be wondering why only sex chromosome abnormalities are abundantly reported, right, compared to the autosomal chromosome abnormalities. And this is because of the obvious abnormal phenotype observed associated with the autosomal chromosome abnormalities. So usually when you have a newborn fetus or animal having this numerical autosomal chromosome abnormality, usually it will come out with a developmental defect. So the farmers will be able to detect easily, and they will eliminate, or will be culling it from the herd without being tested, right? So because of that, we don't have much of the reports from those kind of abnormalities. But for numerical sex chromosome abnormalities, as you can see, the external genitalia even may appear normal, right? But people will later on understand that this is having problem in terms of fertility when they are not able to produce new offspring. So because of that, they are usually detected and reported. So for this reason, you can see most of the reports are sex chromosome related abnormalities and numerical errors, all right? So as a result of this, my conclusive comment is we need to make sure those breeder animals to be used for breeding to be checked also for these cytogenetic problems, all right, before they are allowed to go for breeding, in order to prevent any harmful effects of infertility, sterility, and so forth within the herd, all right? And animals having fertility problems, but with no obvious phenotypic defects, like carriers, for example, with the translocation 129. We need to also be careful of those kind of animals. And sometimes when you have a fertility problem in the herd, and not related with like very obvious and commonly implicated causes like infectious diseases and nutritional problems, please take into account to consider the cytogenetic investigation to check if there is any chromosomal errors behind the problem. With that, I finish the second part of my presentation, and I thank you very much for your attention. And sorry.
Thank you. Thank you very much, Dr. Nur Hussein. Because the time is because the Maghrib time in our place now is coming. We have to finish without discussion. Okay, I'm so sorry. This is the time. I will return this lecture to the MC. Thank you very much.
Thank you very much to Dr. Chandra for guiding this session. Okay, we go to the next agenda. On behalf of Faculty of Veterinary Medicine, Airlangga University, we would like to hand over the certificate of appreciation to our speaker virtually. For Professor Dr. Tita Damayanti Lestari, Doctor of Veterinary Medicine, Master of Science, the time is yours.
Okay. Thank you, Dr. Chandra, Isara Vita, for guiding us in this interesting presentation by our guest lecturer, Dr. Nur Hussein. Dr. Nur Hussein, we really appreciate of your time with us giving this lecture. It is important and it is interesting. Maybe I add a little bit from the first presentation. Yeah, that yes, we think about application of AI in small ruminant by you don't have to ask, you don't have to answer by laparoscopy, right? Because alumni who live in Malaysia, Faris Pakar has Maiternak. Maiternak is small ruminant farm, and he also applied AI by laparoscopy. Yeah. And he is success.
Very good.
Yeah.
Interesting.
I'm not remember which part of Malaysia, but he is in Malaysia, and we also invited him last time. And the second presentation, it is important, yeah, that we must consider for the cytogenetic check. Okay, thank you very much for your to avoid the problem. Okay. Once again, thank you very much, and on behalf head of the division of veterinary reproduction, I hereby submit the certificate for you virtually. So, Saudara Fajar, can you help me? Yeah. Okay.